Hello students, welcome to Livy's Academy, your educational catalyst. In this video, we will be discussing about the G protein coupled receptors. Whenever we study about cell signaling or any other signal transduction pathways, we come across with this term called as G protein coupled receptors. So what are G protein coupled receptors? G protein coupled receptors are largest family of cell surface receptors. So what are cell surface receptors? These are the receptors which are found on the surface of the cell that can bind with a ligand and pass the signal to the cell. We have already uploaded a video on the basics and introduction of cell signaling. So before watching this video, if you are unaware about the basics, please go and watch the basic videos. Coming back to GPCRs, these are the receptors coupled with a G protein, and the G protein is located intracellular. So that is why these receptors are called as G protein coupled receptors. GPCRs are members of a large family of proteins found in eukaryotes and in some certain prokaryotes also. They were discovered by Robert J. Lefkowitz in 1970, and the structure and function were elucidated by Brian K. Kobilka, and they were awarded the Nobel Prize for Chemistry in 2012. A remarkable property of GPCRs which make it distinguishable from other classes of receptors is the presence of 7 transmembrane alpha helical regions, so that they are also known as serpentine receptors. As you can see in this figure, they are transmembrane helices which are oriented perpendicular to the plasma membrane. The major functions of GPCRs are the growth and proliferation of cells, chemotaxis, neurotransmission, synthesis, etc. Members of the GPCR superfamily share the same architecture, that is 7 transmembrane alpha helices, an extracellular amino terminal segment, and an intracellular carboxy terminal tail. The 7 transmembranes will be connected via 3 intracellular loops and 3 extracellular loops. For example, this is the plasma membrane. And the N terminal is extracellular and the C terminal is intracellular, and there will be 3 extracellular loops and 3 intracellular loops. Here the third intracellular loop is unique in the diversity of its sequence and also in the length compared to the other loops. G protein coupled receptors are the largest class of cell surface receptors with more than thousands of GPCRs identified so far. So there are mainly 6 families in G protein coupled receptor superfamily, that is they are family A to family F. Family A, that is the rhodopsin receptor family, and the main ligands in this family are odorants and other small ligands. Coming to family B, that is the secretin receptor family, here the ligands are high molecular weight hormones. Family C consists of the metabotropic glutamate receptor family. Here GABA B, taste, smell, etc. are the ligands. The next one is fungus pheromone receptor family. Here the ligands are some pheromones associated with GI, that is inhibitor G proteins. And the next one is the CAMP receptor family. CAMP, as we already studied, it is one of the major important secondary messenger in cell signaling. So in here it is usually found in Dictyostelium discoideum. And in family F, that is the frizzled or smoothened receptor family, it is usually involved in the cell polarity segmentation, etc. So I have already mentioned that G protein coupled receptors are the receptors which are coupled with a G protein. So that is the important thing in this receptor, G protein. So what is G protein? G protein means guanine nucleotide binding protein. So these proteins are belonging to the GTPases. So usually these are the important molecule that transmit the signals from outside of the cell to inside. So they belongs to the GTPases, that is the largest group of enzymes that can hydrolyze GTP. That is why it is called as GTPases. Every G protein has an intrinsic GTPase activity so that they can switch off themselves. So we will be discussing it in detail, and these proteins are activated when the bound GDP is released and a new GTP is added. So usually the G protein will be in an inactive condition with GDP. So whenever this GDP is released and GTP is added, it will get activated. For this exchange reaction, there requires an external protein called GEF, that is guanine nucleotide exchange factor. So here we can see that whenever GDP is bound, the G protein will be in an inactive conformation. That is, there is no signal, GDP is already bound, it is an inactive conformation. Whenever a signal comes, the GDP releases and the GTP bounds and it will be activated. That is an active conformation. After a short time, they hydrolyze the GTP and come back to their inactive state. Coming to the structure of G protein. G protein has two domains, GTPase domain and helical domain. Coming to GTPase domain, this is where the GTP binds and gets hydrolyzed. It's also called as P-loop. Inside the active site, the GTP and the water residues are misaligned. So here the GTP hydrolysis is slow. The rate of GTP hydrolysis determines the duration of the signal. And second one is helical domain. This is a variable region which functions to deeply bury the bound GTP in the protein. The rate of GTP hydrolysis can be increased by external proteins called GAPs, that is GTPase activating proteins. The GAP contains a critical loop called as arginine residue. When a GAP binds to a G protein, the loop is inserted into the active site. So the arginine coordinates with the GTP and decreases their negative charge, which will further increases the rate of hydrolysis. So when the gap is on, the GTP hydrolysis will be fast and there is short signal duration. G protein is a trimeric protein with subunits alpha and beta gamma. So here, as you can see in this image, in the absence of a ligand, the G protein is in an inactive condition. In the inactive state, G alpha is bound to G beta gamma dimer and GDP. So only when a ligand comes and binds with the receptor, the G protein gets activated. So what are the ligands that are coming and binding to the receptor? They can be hormones, neurotransmitters, adrenaline, taste, etc. So usually GTP will be present inside the cell. When a ligand comes and binds with the receptor, the activated G protein coupled receptor catalyzes exchange of GTP for GDP on the G alpha subunit. As a result, conformational changes take place in the G protein coupled receptor, which leads to the dissociation of G beta gamma dimer from G alpha and thus activate multiple molecules of G proteins. The activated G alpha and G beta gamma proteins in turn binds to various effectors. The alpha subunit is a GTPase and once it hydrolyzes its bound GTP to GDP, it reassociates with a beta gamma complex to the inactive G protein. Two important signal transduction pathways involving G protein coupled receptors are CAMP pathway and IP3 pathway. The G protein activates adenylyl cyclase which converts ATP to CAMP. CAMP further activates enzymes like protein kinase A. Coming to the IP3 pathway, IP3 is inositol 1,4,5 triphosphate, which is released to the cytosol as a result of phospholipase C. The IP3 will further bind to ligand-gated calcium channels and helps in the release of calcium ions. We have already discussed the CAMP and IP3 pathways in detail in our previous videos, so please go and check those videos. Link will be provided in the description box. Another G protein called as inhibitory G protein, that is GI, inhibits adenylyl cyclase. Both G proteins and inhibitory G proteins are targets for some medically important bacterial toxins. For example, cholera toxin, which is produced by the bacterium that causes cholera, is an enzyme that catalyzes the transfer of ADP ribose from intracellular NADPS plus to the alpha subunit of G proteins. This ribosylation alters the alpha subunit so it can no longer hydrolyze its bound GTP, causing it to remain in an active state that stimulates the adenylyl cyclase indefinitely. This results in the prolonged elevation in cyclic AMP levels within the intestinal epithelial cells, and it will cause a large efflux of chlorine ions and water into the gut and thereby cause severe diarrhea that is characteristic of cholera. G alpha has an intrinsic GTPase activity. We have already discussed that. The G alpha bound GTP complex is the active one and it diffuses it to activate target proteins. The major effector proteins are adenylyl cyclase, phospholipases, etc. And it usually increases the CAMP and calcium ions, cadherins, etc. and activates CAMP, CGMP gated channels also. Coming to G beta gamma complex. It was initially thought that the beta gamma dimer complex acts as a negative regulator and it blocks adenylyl cyclase. But later it was discovered that it has also an activation property. It activates muscarinic K plus channel, phospholipase A2, phospholipase C beta, PI3K pathways, beta adrenergic receptor kinases, some ion channels, etc. Now it is clear that many effectors are regulated by both alpha and beta gamma subunits. So that's all about the basics of G protein coupled receptors. Stay tuned for more videos on cell signaling and signal transduction pathways. Thank you for watching. Please like, share and subscribe. Like is Academy, your educational catalyst.